And okay, so today I'm going to be talking about some of the research I've been doing. I'm in actually two labs, Albert Xiao's and Tom Liu's. So this is my work with Albert Xiao, and it's basically simulation of MRI case-based cardiac motion corruption for deep learning-based artifact detection and correction. So I'll explain what all that means. Um, so basically, first, cardiac MRI. It has it has excellent soft tissue contrast and high spatial resolution, so it's becoming like the premier imaging modality for cardiac. In addition to providing like highly detailed structural information like you see here, can you guys see my mouse? Oh, cool. Yeah, like you see here, it can also provide functional information, which can be used to diagnose and assess numerous cardiac conditions. So like 4D flow, for example, you can see hemodynamic information. And you can also get strain and strain rate information, kind of small, but from SSFP MRI. So all these things have been shown to help with assessment and diagnosing different types of cardiac diseases, which other imaging modalities can't really do. So there's a lot of plus sides to MRI, especially for cardiac. But unfortunately, there's no such thing as free lunch. So MRI suffers from poor temporal resolution, which leads to motion artifacts. So we get all these awesome, like quantitative information from MRI, but it's often corrupted by motion. And so a big part of what MRI researchers focuses on is how can we, get around motion. Also, how can we also see how motion is occurring for the heart? Because heart is something that's moving. We want to see how it moves. But we also don't want like a bunch of blurry images. So to explain more about what this motion does, maybe I thought of a good example is just motion in photography, something we're all familiar with. So to help me with that, I brought a friend. This is Remy. Remy likes to move a lot. And so what you can see in this first image is there's no motion. He stood still for once in his life. There was a little motion, especially around the pause. You can see how it gets a little blurry there. And then, well, you can barely tell what's going on here. So just like that for photography, you also have a similar problem with cardiac MRI. If you have motion that looks more like this, it's really hard to tell what's going on, making it not, good enough for like diagnosing someone or assessing their condition. So these are some of the motion artifacts you see with MRI, and they look pretty bad because it isn't just people moving that causes problems. I mean, it's also just your heart moving and your breathing, something you really can't control. And it can cause motion artifacts that look like this, making it very difficult to tell what's going on or that this is even a heart. And so making it really challenging for people to use this information and diagnose. So trying to find ways to mitigate this is a big part of a lot of people's research in MRI. So deep learning is the hot new thing right now. And it too can be used to help correct for this, but there's a bit of a caveat. So it can correct for motion artifact. It also can, well, we're also supposed to show that it can do a pretty good job of quantifying motion severity. So why might we want to do that? Well, it can be just, maybe we can't correct the motion, but it can tell maybe the technician that they need to repeat the scan. Because it's not always clear, like, is it too bad, or can we live with it? And then also it can inform like radiologists, researchers of the severity of motion artifact in an image, which may change how the image is interpreted. And then lastly, it also could be used to measure the effectiveness of various motion correction methods. So if we could somehow quantify the motion severity, then if we make some way of fixing it, we could see how well it fixed it, right? But in order to use deep learning, we have to be able to generate a training set. We don't really have a ground truth because we don't really know what it was supposed to look like without the motion, if we can't get it without the motion. This is where the challenge, this is where it becomes challenging. A lot of people who have done deep learning type methods for correcting and quantifying motion severity, they often just like score it. So they have a radiologist come in and score the level of motion, which is very subjective to the radiologist. And it's no way a perfect way of doing things. So what if we could somehow create the motion ourselves, something that's realistic, and we can use that to train, because if we can create the motion, then we know what the original ground truth looked like, the good image, we create motion on top of it, and then we can use that to train a neural net, so we know what the ground truth is. But doing this can be challenging because how do we make it look realistic? So, for example, what if we took a bunch of different motion states like what you see here? If we could somehow combine it into one image, then that would be essentially what that motion blurring is doing, right? Like when my cat was moving really fast, it was because you saw it was taking data from a lot of different motion states all at once. And that makes it difficult, or that's what gives you that kind of blurring look. So if we can kind of create it ourselves, then we have now like a synthetic motion for our training set. And I basically just said that. Okay, so side note, but you don't have to think too hard about this. MRI doesn't sample like a regular like camera does. It actually samples in K-space, which is just a Fourier transform. And so basically it reads in like frequency information of your image. And it does it one line at a time. So if the person is moving, and for the first line of K-space, they're in one motion state, and for the second line, they're in a different motion state, that's kind of like what we would assume the motion, that's what's going to affect the image once we do the inverse Fourier transform. So, but maybe it's probably better just to think about like the original example here, especially if you're not used to Fourier transforms, but it's kind of the same idea. So one great thing about MRI is cardiac MRI is they came up with this way of actually getting different motion states. It's one of the methods for correcting motion. It's called cine MRI. Basically, what they do is they take a lot of data and they look at your ECG over the times you took that data. And they take the individual samples from each line of that K-space and they bin it into motion states. And that effectively gets you multiple motion states. But granted, it occurred over multiple cardiac cycles. So it's not like, we managed to actually make the person still. It's just that we separated the data based off what motion state we believe they were in. And it does a pretty good job, but it means that you have to scan a lot longer and you can't get a lot of that quantitative information that you want because you're basically breaking up your data. It's not really that continuous. So in order to do this, first step was we developed a way of how can we shuffle that information into one image. And we did that by basically parameterizing this problem. So I thought of the best way to show this was probably through an example, and I'll just jump in. So let's say we have multiple different motion states of K-space. So each one of these, the person was in a different motion state. The first thing that we want to do is if I want to corrupt the very first state, I'm going to have to take, maybe 60% of that image and get rid of it and take stuff from other states and put it in its place. So that's the first thing. That's the first parameter that I have is how much do I take from my original image and replace that with the other images. The next, that was basically that. And then next, what I do is I figure out which part of the state or which images I actually take from. So for example, if I take from the image that's right next to it, there probably isn't a big difference between the motion. If I take from further away, like maybe the difference between systole and diastole, you'll see a big change. And we want both because we want to be able to quantify both very little motion artifact and a lot of motion artifact. Then the next parameter is how many do I shove into that one state? How many different motion states do I combine together? That's what the beta character does. That's also something that we change in our algorithm. And then lastly, we determine basically which part of K-space we remove. So we could have removed any part of it, and different parts of K-space change the image in different ways. Particularly the middle of K-space actually changes more of like your low frequency signals. So changing stuff there means big artifacts. Changing stuff on the edges means very little artifacts. So this is kind of our way of being able to get a wide range of artifacts without actually having that much data. And here's kind of like an example of how I took state 0 and made it into a motion artifact, or corrupted it with motion artifact from state 9, 10, and 11. And so this is kind of what it did. So here you have the cardiac SSFP sequence. So this is Cine, and it has, I think, 20 motion states. And what basically I've been able to show is that I can make varying levels of motion artifact and I can control it. So because I have this parameterization that we did, I now have a way of tuning these parameters to get pretty much any level of artifact I want. And this is becoming very useful because now I could train a neural network. So you can see here's the mild, here's moderates, and here's more severe. You can see like this shrieking kind of looking thing. All right, so now we have to go to the fun part, the neural network. So basically what we did was we had an architecture that was like kind of multitask. So one, we have a unit that's for the motion correction part. So what we're feeding in is this synthetic image. What we want out is the ground truth image. So we give both for the training. The second thing, in order to make this a little bit better, is we also gave it that score. So I didn't mention, I kind of jumped over it, but the score that I gave it is the SSIM 1 minus it. So SSIM is the structural similarity index measurement, and basically it tells how much the two images are alike. Mainly structural, but also like intensity as well. And so a score of 0 here, because it's 1 minus it, would mean that there's no difference. These images are exactly the same. So we give it both. So we want it to not only correct the image, but we also want it to score the original artifact. How bad is it, essentially? From there, here are some examples of what the neural net was able to do. So on this side is the original, like the synthetic image. So this is where I corrupted it. So we have motion artifact. Here's the ground truth image without the artifact. And this is what the neural network popped out. So ideally, if the best case scenario is we want the corrected image to look like the ground truth image. And it's starting to do that. It's a little hard to see on the screen, but there's some ghosting here that got removed in this image. There's still a little bit of artifact, but it's getting much closer to this. Also on top of it, this is supposed to say SSIM, sorry. The ground truth score was 0.9221, which basically says that like this is how bad the original image was, but the corrected image has a 0.955, so it got better. That's what we want to see. Here's another example, which I thought was nice. So where the arrows are pointing are some of the areas of the artifact in the synthetic image. You can see that it's pretty much gone in the corrected image, which is kind of what we like to see. So I like this one a lot. And it looks a lot more like the ground truth image. And you can see that again by the SSIM score. So the ground truth, this is like the original difference between the synthetic and the ground truth image. The predicted is what the neural network said it was. So basically, like just looking at this one, can it tell me how bad it thinks it is? And so it got pretty close. And then the last one is the corrected. So it got a lot closer to 1, which means that it's doing its job and it's making it look a lot more like the ground truth image. So this is just the training set, right? Like, because I generated these images, these synthetic images, they're not the, the images that we're going to actually put in for a test. So to do that, we actually had to get a radiologist. So we got an expert radiologist to grade a different set of images on how bad they were. So the different classes here, class 0 to class 3, are the level of severity. And so class 0 is like very little motion artifact. Class 3 is a lot. And the results are kind of iffy, but we can explain why. And we have actually corrected it since then. But basically, what you're seeing here is that this on this side is the predicted SSIM value from the neural network. And this is what the radiologist said was how severe it was. So ideally, we would want these 4 box plots to be kind of separated so we can make it separable. So we could say, oh, okay, if I see an SSIM score or a predicted SSIM score of 0.99, then and up, that means that it's good, it's class 0. But anything under 0.80, it's bad, it's class 3. That's what I want to be able to see. But as you can see here, that doesn't do such a great job because especially class 3, it's all over the place. So I'm going to tell you why on the next slide, but just keep that in mind for a second. Other things I wanted to show was here's an example of maybe that one case we're just telling a radiologist scan again or you're fine kind of example. So here is like for the good case is class 0s 1 and 2. If you get those, then you can keep going. You don't need to rescan. But if you get class 3, you have to rescan. And it was able to actually get a pretty good threshold on where that would be. So it would be a predicted SSIM of 0.959. And that would actually give a pretty decent true positive rates without having like a horrible false positive rate. So overall, that was good, but we still need to address this problem. And so, as I promised, this is why it occurred. Basically, when you look at these top images, the SSIM score is shown here. It's 0.885. But if I zoom in on the heart, it gets a lot worse, right? Because the way the SSIM works is it's giving an overall how much are these two images alike, and it's looking at the entire image. But a radiologist, when they look at the image, they really only look at the heart itself, and they don't look much around it because that motion isn't really affecting them. And so that's why when you had these original classes, why they were all over the place, because basically they weren't really looking at the rest of the image, that radiologist, they were only looking at the heart. And so when I redid these SSIMs with basically a cropped image, the data started to look a lot more like what the classes were. So now we're retraining. I don't have those results yet, but maybe by the symposium. So we think it will definitely help. And here's another example where it actually got better. So not by much, but for the most part, like you have these two images, they look pretty similar, but when you zoom into the cardiac or to the heart, you see that they're even more there's barely any artifact at all. And so you don't really want to have artifact that's on the edges around far away from the heart, especially to be affecting your score, because that's not what a radiologist would be considered as a problem. Okay, so also on top of it, we mainly just talked about cardiac motion. So now we're looking into respiratory motion as well. But this became a little more of a problem because we didn't actually have different motion states of respiratory, because most people only do that cine imaging, they do it with a breath hold or the subject's best attempt of a breath hold. So getting different motion states of respiratory is becoming very challenging. We don't have that type of data. So how do we train it? So what we did is I just created deformations of the original image in a motion that looks a lot like how we believe the breathing would affect the image. Now, granted, you can see it's over exaggerated and it's probably not what a human would look like, but we found it actually worked pretty well. It's able to create a pretty good synthetic motion artifact that looks quite realistic to what we see, even though it is a little bit, this probably does not happen. And this makes it really nice because we can apply this to all the images, and now we have the respiratory motion as well. So here are some examples. So again, here's you wouldn't really see the heart move this way, but it gets really what you want to see is that border move, the edge of the abdomen, and the chest wall is what you want to see move, and that's what's moving. And you'll see that type of artifact up here. A little hard to see, but basically these lines, especially around here, we see that a lot in cardiac MRI. So it was able to create that. It was also able to score it pretty well as well. So this is on this side is the least severe all the way to the most severe. And then also here, another example, this one did a pretty good job as well. You can see that we really have a lot more cardiac motion and the whole chest motion as well. So that means that we can get much more severe motion artifact, which hopefully will help our algorithm better handle different types of input, because we only gave it basically cardiac motion. If it sees respiratory motion, it's not going to perform as well. So that's our next step. So in conclusion, realistic motion corruption can be synthetically generated by applying this case-based swapping thing to MR Cine data. A wide range of motion severity can be simulated using case-based swapping to better train deep learning-based artifact detection and correction methods. And then our future work, we're currently training the model right now, is to utilize a smaller cropped image right around the heart to improve the performance so that the SSIM doesn't isn't dependent on motion that's occurring farther away from the heart. So hopefully we'll have those results in the next few days. And then also we want to train the unit on both the respiratory and the cardiac data, or cardiac motion data, which is currently also being done. So we're having results that are hopefully going to be popping out pretty soon, and we're pretty excited. So, and then lastly, this kind of a side project, but we're trying to also adapt the pipeline for 3D abdominal imaging because they really suffer from motion artifact big time and making it very difficult to get like pretty much all abdominal scans are 3D, and 3D is even worse because you're trying to collect a lot more data and the person doesn't stay still at all. So that's the next step as well. And so this is my lab, and yeah, thanks for listening.
Questions?
Yeah.
Great talk.
Thanks.
I was just wondering, like, when you do your cropping, is there a way you like quantitatively decide how you're going to crop it?
Yeah, so there's probably so we were thinking about doing like a mask, which there's a lot of methods for masking like the heart. But we decided that heck, the heart, I mean, is really not going to be much bigger than 16 centimeters by 16 centimeters. And usually, actually, pretty much all technicians are going to put the heart in the center of the image. And so we just tried it and it worked like on I checked every single like subject and it perfectly like cropped it very well. So it basically just took the center 16 centimeters in both directions and called it. And so that's like the naive approach that actually happened to pan out for once. But our idea was that if it was a problem for some, we would have done a masking type thing. But we didn't have to. So far, so good. Yeah.
Can you tell me more about, I'm sorry, first of all, like, great.
Thanks.
Super cool. Can you tell me more about the training process for the neural network? Like,
Yeah, sure.
how many, like, so each sample was like a 4D video, right? Like you have.
Yeah, okay, so it's a lot. So probably like just to give you like a ballpark number, it's probably 100,000 images. But what that comes from is I think there is a total about 30 subjects. From these 30 subjects, they probably had about 10 different imaging, like we call them series, just basically different views of the heart. And each of those views of the heart, we have multiple slices. So there's probably for each one, maybe 5 to 10 slices. So for every single one of those, there's like 20 like cardiac phases. That means like cardiac motion states. And we take that and we do that parameterization thing. We basically shuffle them around and we generate about up to 50 images of each motion state. So we had a total of motion severity. So we had a total of 10 bins. This was like the SSIM bins, and each bin was like a range of SSIM scores. They could have up to 50 images in each one. That's why we had so many, because it was like one person's image could have created one person's data set could have created 500 images, because I can basically create different levels of severity of that same image. That's why it's like we have so much data, basically. Did that answer?
Yeah, totally.
Cool.
And then once you've done the image correction, how do you like ensure that the corrected image is giving the physician the information they need to know, right? Like,
yeah, so basically our idea for that part, so we only could correct obviously to some extent. Like, well, I guess our idea is basically this. If you remember, I showed this plot. It's basically saying that if it's still categorized as class 3, which we gave a threshold for the SSIM for that, then it's still bad. So ideally, if we corrected it and it's still under that threshold, then we would say that this is probably not good enough. Now, granted, that doesn't mean that they still can't get that information. It's just based off for our data that's generally what it looked like, based off that one radiologist. So ideally in the future, we're going to have probably 5 to 10 radiologists score it, and actually see if that holds across it. Yeah.
okay. No, okay, so I think that makes a lot of sense. More broadly, like one of the things I wonder with these image correction methodologies is that that it makes the image like look better.
Yeah.
But that's different than if
yeah, basically. Yeah, so you're right. Yeah, so basically I can make it look good, but it doesn't mean it was the right data, right? It just means I made it look more like what it thinks it should look like. So for at least the training purposes, we do have the ground truth. because I had the ground truth image that I then injected like the synthetic motion. So I know what it should look like. Unfortunately, we still don't ever know what it will look like for the test data set, and there's nothing we can do about that. Best case is that what we can do is we can also get like anatomical images. These take longer to acquire. So sometimes their motion artifact is like not so great either. But if we do like the cine idea where we just acquire for a very long time and bin it into motion states, then we can look at that and compare it to the two to actually make sure, because those ones tend to have, you know, motion correction already built in. It's just that that doesn't work for every type of imaging that you need to do for cardiac MRI. Like all the stuff with the quantitative stuff, you can't have that. You need to be imaging much faster. So that's probably what we'll do to at least give us some confidence, but there's nothing like, there's no perfect ground truth for the test data.
Yeah, no, that makes a lot of sense. Thank you, Noah. This is super cool. Really
Thanks. Yeah. Anything else?
Okay, I'm gonna pause it here.
Cool.
Hi everyone, I'm Samantha. Nice to meet y'all. I'm excited to present my talk today, entitled Engineering Chemogenic Biosensors for Highly Sensitive Interrogation of Spatially Regulated Cell Signaling Events in Living Systems. So in my talk today, I'll talk about how and why we engineered chemogenic kinase biosensors, how we've applied chemogenic protein kinase A or PKA biosensors, and how we're expanding these biosensors to other kinase targets like the calcium calmodulin dependent protein kinase 2 or CAMK2 for in vivo imaging. So why do we need biosensors anyway? Well, biochemical signaling regulates everything that our cells do, everything from gene transcription to metabolism to migration. But how do our cells know when and where to perform these different functions? Well, we know that cell signaling is spatially regulated, and it's this spatial regulation that dictates when and where these events occur. So we can use biosensors to look at the spatial regulation of cell signaling in real time. Biosensors usually have a sensing unit for a given target of interest, as well as a reporting unit. And in many kinase biosensor designs, the sensing unit will have a phosphorylation substrate and a phosphoamino acid binding domain. And the reporting unit will have Forster resonance energy transfer or FRET compatible fluorophores. So phosphorylation of the substrate can cause it to bind to the phosphoamino acid binding domain, inducing a conformational change that changes the FRET efficiency of the two fluorophores, giving you an optical readout of kinase activity. But the problem with a lot of these FRET-based biosensors is that they're not ideal for multiplexing or in vivo imaging, just because the two fluorophores for the reporting unit are usually occupying two different and often blue shifted imaging windows. And so we overcame this with a new kinase biosensor design that uses a chemogenic reporting unit. Chemogenic reporters combine the subcellular targeting capabilities of genetically encodable reporters like fluorescent proteins with the flexibility of tunable and photostable chemical reporters like dyes or stains. In our sensor's chemogenic design, we use a genetically encodable self-labeling halotag 7 protein in a circular permutation. And this halotag protein can incorporate a chloroalkane modified dye through an irreversible dehalogenation reaction, and this covalently links itself to the dye that fluoresces upon excitation. So to create a chemogenic protein kinase A biosensor, we sandwiched a circularly permuted halotag reporting unit between a substrate sequence and an FHA1 binding domain. We then labeled this chemogenetic halotag-based A kinase activity reporter, which we called Halo ACAR, with the far red fluorogenic dye JF635. When the substrate is phosphorylated by pKa, it binds to the FHA1 domain, causing a conformational change in the halotag, and this produces a change in the fluorescence of the conjugated dye. When we tested this sensor, we expressed it in HeLa cells and imaged in the far red channel, and we then stimulated pKa activity with the adenyl cyclase activator for scolin and the phosphodiesterase inhibitor IBMX, and then inhibited pKa activity with the H89 ATP analog. And in doing this, we achieved a more than twofold increase in the fluorescence intensity upon maximal stimulation compared to the kinase dead control, shown here in blue. And this response was reversible upon the addition of H89. We also tested the specificity of the sensor for pKa by activating and inhibiting PKC. We saw no response in response to PKC activation, so that tells us that what we were seeing was selectively reporting pKa activity. From this, we optimized the dynamic range of the sensor through both rational and library screening of the linker sequences between the sensing unit and the reporting unit. So after doing this, we constructed sensors with 3 new linker combinations that produced high basal brightness with lower dynamic range, moderate basal brightness and dynamic range, and low basal brightness with very high dynamic range. These are called Halo ACAR 2.0, 2.1, and 2.2, respectively. And when we tested these sensors in HeLa cells, we found that we achieved a more than 12-fold dynamic range with the Halo ACAR 2.2 sensor, and this drastically outperformed any far red protein kinase A sensor available. And with this new suite of chemogenetic pKa sensors, we were able to image pKa dynamics in more complex imaging applications that span multiple spatial scales. So of course, we were able to resolve spatially regulated pKa activity at the single cell level. Here, we demonstrated that Halo ACAR 2.1 can show us real-time changes to pKa activity, as we saw both transient changes to pKa activity in response to isopaternal treatment and sustained responses to forskolin and IBMX treatment in individual HEC293 T cells. And next, we wanted to see if we could image pK activity at specific subcellular locations. So to do this, we tagged our biosensors with localization sequences for the locations of our choice. So here I'm showing the sensor targeted to beta-actin, and we observed that the sensors still reported changes to pK activity, largely following similar dynamic range and basal brightness trends among the different linker variants that I mentioned earlier. And still in the train, subcellular targeting, we also targeted the sensor to clathrin-coated pits. These pits are typically on the submicron scale, so they're usually hard to resolve with just confocal microscopy. So we moved into the sub-diffraction limited scale and imaged the sensor at these pits with STED or stimulated emission depletion microscopy. And even in the unstimulated state, our sensors were bright and photostable enough to be imaged with STED, but more optimizations would need to be made for time-lapse microscopy of the sensor with STED. But pivoting from imaging in smaller scales, we also imaged in larger scales. We generated 3D sensor expressing cell spheroids that were around 60 microns in diameter after the 4th day after seeding. And we observed responses to PA stimulation and inhibition in this 3D multicellular scale that were similar to those that we saw on the 2D scale. And although we saw these responses, we wanted to see if we could better resolve the individual cell boundaries here in the Z-plane. And so again, we tried imaging the 4-day cell spheroids, but this time with 2-photon microscopy. And we saw that we were able to resolve the individual cells in the middle of the spheroid. But we also found that our collaborators' multiphoton laser could only achieve partial excitation of JF635 since it only went up to 1100 nanometers. So what we did is we saw that this inefficient excitation appeared to limit the dynamic range that we saw in the sensor to only approximately a twofold change compared to the sevenfold that we expected from the sensor. So to address that, we took advantage of the sensor's spectral tunability and labeled the sensor with JF585 instead, which is a red dye. We saw a greater signal from the sensor in both the unstimulated and stimulated states. And we also achieved the full anticipated twofold dynamic range of the sensor labeled with JF585. This can be very useful for multi-photon imaging experiments, especially when imaging on different microscopes. So I know a lot of the labs that I've seen imaging tissues with multi-photon microscopy, they already have red and green channels set up. And so this kind of shows that these chemogenetic sensors can be already accessible for labs that have those channels set up. And so after this, we wanted to see if we could achieve higher resolution 4D imaging of the entire spheroid volume. So we worked with the Schoenberg lab. Here you see San Diego to image these spheroids with lattice light sheet microscopy, and we were able to image the entire volume of our spheroid with very good spatial resolution, as you can see from these 3D reconstructions of the spheroids. And while these frames were captured at very high spatial resolution, we saw losses in sensor signal when trying to acquire time-lapse measurements on the lattice light sheet microscope. So again, we still need some optimization to image time-lapse images for high-resolution 4D PK imaging, but we still were able to see a change in the sensor with this great spatial resolution. That being said, we still wanted to push the capabilities of our sensor and image PK activity on the tissue scale in isolated mouse pancreatic islets. So we imaged glucose-starved islets expressing halo-ACAR, and then adding glucose resulted in changes in fluctuations in PK activity. And since glucose induces calcium spikes and pKa is involved in an oscillatory circuit with calcium, we're pleased to see that our results corroborated these models and that halo-ACAR could actually give us a way to directly interrogate PK activity in these living islets. So building on these results, we wanted to also branch out with our design to see if we could successfully generate chemogenic sensors for other kinase targets. And so since biosensor design is very modular, we found that we were able to adapt the halo-ACAR design by switching the substrate and phosphoamino acid binding domains in the sensing unit, and we were able to create chemogenic sensors for protein kinase C or PKC, protein kinase B or AKT, and extracellular signal regulated kinase or ERK. And we saw that these new sensors maintain similar behaviors between the different linker variants. Of these new sensors, I wanted to highlight one of our new very promising chemogenic biosensors for calcium calmodulin dependent protein kinase 2 or CAMK2. So CAMK2 is a valuable biosensor target because it's important for dendritic spine function in neurons. It can help to regulate NMDA and AMPA receptors, and as well as spine formation and dynamics. And both of these things are crucial for synaptic plasticity and long-term potentiation. Taking a closer look at CAMK2 structure and activity, you'll usually find it as a holoenzyme with 12 subunits, and each subunit will have a catalytic or a kinase domain, a regulatory domain that has a calmodulin binding element, and an association hub that allows it to come together with other subunits to form this bigger holoenzyme structure. Prior to activation, the regulatory segment of each subunit is bound to the substrate binding groove of the kinase domain and suppresses catalytic activity. And once it's bound by calmodulin, it releases from the kinase domain and allows catalytic activity to occur. Once the regulatory segment's unbound, there are specific threonine residues on it that can be phosphorylated to enable autonomous CAMK2 activity that's independent of calcium or calmodulin binding. Phosphorylation at threonine 286 prevents rebinding of the regulatory segment to the catalytic domain and prevents autoinhibition of the enzyme. Whereas phosphorylation at threonine 305 or 306 blocks calmodulin rebinding. And so with this, CAMK2 can enter its calcium independent and autonomous state. And this allows CAMK2 to transduce highly transient calcium signals into longer lived signals. And it's thought that this autonomous state is important for long-term potentiation and memory. And so most existing CAMK2 biosensors are based on a sensor called Kamui alpha, which uses a full-length CAMK2 alpha subunit as its sensing domain. And when we test these CAMK2 biosensors, we usually elicit changes to the calcium levels in the cell to simulate CAMK2 because of its calcium-dependent nature. And so stimulating with thapsigargan and alleomycin releases calcium stores and creates a transient spike of calcium in the cell, whereas adding calcium chloride produces a more sustained increase. And because of CAMK2's calcium-dependent activity, we expect the CAMK2 biosensor to closely follow the calcium biosensor trace, which I'll usually show on the right side, y-axis, and then compared to the CAMK2 biosensor trace, which I'll usually show on the left y-axis. So while Kamui alpha appears to capture CAMK2 activity with these simulations, overexpression of full-length substrates can artificially affect CAMK2 signaling. We don't want that. So a recent biosensor called Fresca overcame this limitation by using a Syntide2 substrate peptide and an FHA2 phosphamine acid binding domain as its sensing unit. But the thing is, with this substrate-based sensor, is it had a very low dynamic range of only around 2%, and it also had a very noisy signal. And we also found out that the Syntide2 substrate can also be not specific for CAMK2 and report protein kinase D activity. So to address these limitations, we engineered a CAMK2 biosensor using a new CAMK2 specific substrate, which we called CAMK2 activity reporter, or CAMCAR. And CAMCAR showed a dynamic range of around 80% change in response to CAMK2 stimulation. And we also saw that CAMCAR did not respond to stimulation when a CAMK2 specific inhibitory peptide was also co-expressed. So it showed that it was specific for CAMK2 activity. And while this CAMCAR already showed a 40 times improvement in the sensor response compared to Fresca, we wanted to generate a more sensitive single fluorophore version that could be used for more complex imaging applications. And so we wanted to create a chemogenetic CAMK2 biosensor, which we would call Halo CAMCAR, by replacing the pKa substrate with our newly engineered CAMK2 substrate. So to our excitement, this gave us a very sensitive chemogenetic CAMK2 sensor, and Halo CAMCAR responded with a very high dynamic range of nearly 30-fold change, or around 3,000%, upon CAMK2 stimulation compared to the kinase dead control here. And I'd like to note that this nearly 30-fold dynamic range is like the highest biosensor range that we've heard of to date, which makes it really sensitive to small signaling changes. So we're very excited for this. We also confirmed that it did not respond to protein kinase D stimulation, so it showed that it was specific for CAMK2. And then when we made halocam cars with the linker sequences that I talked about previously, it appears that they do follow similar trends for the dynamic ranges and relative basal brightnesses, with halocam car 2.0 having approximately 3-fold dynamic range, 2.1 having approximately 10-fold, and the 2.2 having the nearly 30-fold dynamic range. So we're very excited about these results and these sensors, and we're currently working on a lot of applications for them, but one application that I'm particularly excited for involves calcium and CAMK2 multiplex imaging in vivo. Because of the chemogenic single fluorophore nature of these sensors, we can image CAMK2 in spectrally distinct windows from other existing calcium reporters. So here I'm showing halocam car 2.2 labeled with JF635 and the calcium sensor GCAM3. But if we want to image these sensors in vivo, we might want to consider optimizing the 2-photon excitation of our CAMK2 sensor like we did for Halo ACAR. So when we labeled halocam car 2.2 with the red fluorophore JF585, we saw that while it has a smaller dynamic range than when it's labeled with JF635, it still has a 10-fold change, which I think is pretty great. And it's also notably brighter in the unsimulated state compared to when it's labeled with JF635, even with on-peak excitation for both. And so this can help us get higher signal to noise in vivo while still maintaining a redshifted wavelength for more light transmissivity and less phototoxicity in the tissues. And I'm currently working on implementing this in vivo co-imaging system in Drosophila, but for this talk, I wanted to leave you guys with just a few key takeaways. So we developed chemogenic biosensors with halo tag-based reporting units. Halo ACAR enabled imaging of live pKa dynamics across scales from the sub-diffraction limited level to the tissue level. HaloCar's spectral tunability allowed for efficient multi-photon excitation. We adapted the chemogenic halocar design to image the activity of other protein kinases like CAMK2, and halocam-car achieved the highest kinase biosensor dynamic range to date with a nearly 30-fold change upon maximum stimulation. And so I want to thank the lab and people in my lab who worked with me on this project, thank funding sources, including this training grant. So I'm very excited for your questions. Thank you for your attention.
I do have questions, but I have to run, so I'm gonna email you.
Sounds good.
Thank you.
Thank you.
I did one question out of curiosity. I know, I think Ken sometimes it's used like AlphaFold in, I'm not sure it's designing the linker regions or just as part of the biosensor. But it's all based on like more traditional experimental methods, or were you using those kind of tools for this as well?
Yeah, this was based on more like traditional screening methods. I mean, I don't know if I'd call it like traditional. It's more of like a library screening.
Sure.
AlphaFold, we've tried using AlphaFold to kind of explain some of the changes between the different linker variants that we see from like stimulated to not stimulated states, but without having like crystal structures that really show where the chromophore is, it's kind of hard to draw conclusions from them. Yeah, so we've, I guess, tried a little bit of both, but definitely more of a traditional screening method.
Cool. Thank you.
Yeah.
Anybody else?
Yes.
For your PKA sensors, were there any collaborations that you thought they had like a really cool application of using the PKA that you guys were working with? Like any people you were working with?
Yeah, I would say probably the latest one is like the islet PKA imaging. I think that's more like the differences in like the different cells within the islets, and how they respond to the glucose stimulation and like which ones respond first. They call them like first responder cells. And so like, I guess that's like the coolest, they're working on the different, responses in PA, like compared to the different like calcium responses, but I think it's still kind of getting up on their feet with that.
I'm excited to see your calcium stuff.
I know, right? Thanks. All right.
Anybody else? Thank you, Samantha.
y'all.
Great job.